Hey everybody, Dr. Ollis here. In this short video, we're going to review two of the organic macromolecules in the body, which are proteins and nucleic acids. Some of this will be review from what we covered in class already, so if you want to skip ahead a little bit, you're welcome to. Or if you would like to view this content again, feel free to stick around with me. The first organic compound that we're going to talk about in our video are proteins. Remember that proteins are the second most abundant thing in a cell. The first most abundant, what we have the most of in our cell, is water, but the following close behind that are the amount of proteins that we have. When we talk about proteins, they're made up of several different elements, but the two that are unique to proteins compared to carbohydrates and lipids is that proteins have a lot of nitrogen in them, and some proteins also have sulfur. So a little bit of a different chemical composition than our two organic compounds that were energy sources. When we talk about proteins, we're talking about a polymer or something that's made of many pieces, and those pieces are called amino acids. So we can see over here on the right the structure of an amino acid. It gets its name because it has an amine group here on the left with a nitrogen and two hydrogens, and it also has a carboxylic acid group that we see over here on the right. The amine group and the acid group are what we're going to use to bring together to form what's called a peptide bond, which is special just to proteins. But notice that there's also this area up here called the R group, or you might hear it called the functional group. I like to consider the R groups the personality type of our amino acids, meaning they're going to decide how that amino acid behaves. Does it like water? Does it not like water? Does it want to form a bond with its neighbors? All of that kind of thing, all those characteristics come down to this functional group, this R group. When we talk about building proteins, again, that terminology is peptide bonds. So the proteins used to build the bonds used to build proteins called peptide bonds. We string together amino acids one by one at their amine group and their acid group, and we put them together to build these peptide chains. So I have special names for the number of amino acids that go into each size of a protein. So we have what are called dipeptides when there are two amino acids linked together. That's what we see over here on the right in our picture. We've used the process of a synthesis reaction to build a dipeptide with two amino acids connected to each other. If I were to add another amino acid to this protein that I'm building over here, I'd call it a tripeptide. So tripeptides have three. When we have up to 50 amino acids connected to each other, we would consider that a polypeptide. But once we get above 50 amino acids, and most of the proteins in your body have more than 50 amino acids, now we start to call it a protein. So proteins much larger than a polypeptide, much larger than a dipeptide or a tripeptide as well. For us to be able to use these proteins to do their job, they have to have their amino acids in the correct order, because that correct order is going to dictate the way that those amino acids are shaped. So let's look a little bit at the shape that proteins form into. If you recall from 1441, when we're talking about proteins, they have four different levels of structure. The first level of structure on a protein is called primary structure. Primary structure is the order that the amino acids go in. When I tell you the order of amino acids, what I'm actually telling you is what the identity of these R groups are. So when I talk about alanine versus aspartate versus glutamate, the order of amino acids all comes down to that functional group. A really important idea about proteins is that I must have the correct primary structure for my other structures to be successfully built. So if I change the primary structure, the order of the amino acids, all other levels of protein structure will not work. So one level above the order of the amino acids, the primary structure, is what we call secondary structure. Secondary structure comes in two forms for proteins. The first form is the alpha helix. This is a lot like those ribbons you can buy to put on a birthday gift, so it curls around and around and around on itself. We also have what are called beta sheets. And when we talk about beta sheets, think about folding a piece of paper like an accordion. That's what's going on with my beta sheet structure. Both alpha helices and beta sheets form when I have interactions called hydrogen bonds between the amine group of one and the acid group of another. So these are interactions between the amine groups and the acid groups, just like these were interactions between them, but the difference between primary structure and secondary structure is the amino acids that are interacting in secondary structure are not next door neighbors. In primary structure, they are next door neighbors, which is why we build peptide bonds. If we're a little bit farther away though, we're going to build these hydrogen bonds in secondary structure. I take all of my alpha helices, like I see here, and my beta sheets, like I see here in flat, and I fold them together into a 3D shape. When I talk about the 3D shape of a protein, that's called its tertiary structure. So many of the proteins in the body go up to tertiary structure. They have to have at least tertiary structure to be able to do their job. So the 3D shape of one amino acid chain, of one polypeptide, that's called tertiary structure. Some of the most important functions that we do, though, in our cells require more than one polypeptide chain, and that's what we get quaternary structure with. When I put together more than one polypeptide chain, that's called quaternary structure. So a good example of something with quaternary structure is the hemoglobin protein. It's actually made of 4 different chains that each have their own tertiary structure, but when I fit them together like puzzle pieces, I call that the quaternary structure. When we talk about proteins, it's critical that you know that the shape of a protein, the anatomy of a protein, needs to be correct so that its physiology, its job, can be performed. If I lose my 3D shape, I will lose my ability to function. So there are 2 main structures that we would say with proteins. Some of the proteins in the body are called fibrous proteins. So these are proteins like we use in muscle contraction, or these are proteins like we use to give the shape to our cells, the cytoskeleton. But we also have proteins that we would consider globular proteins. These are the kinds of proteins like hemoglobin that I mentioned before, or over here we see an antibody, which is something that helps protect our body. So we need to have the correct shape, whether it's a Y shape like we see with an antibody, or it's a long skinny shape like we see over here with a fibrous protein. We must have our shape to be able to do our job. If we lose our shape, we're now called denatured. And think about it, if I need my shape to do my job, a denatured protein can't do its job. That's why it's important that we have the correct shape for our proteins. With these 2 groups of proteins, fibrous and globular, I want to briefly compare them to you. But again, I want to remind you that both of these types of proteins have to have the right 3D shape or they will not be functional. So when we talk about proteins, the 3D shape of a fibrous protein is like a strand. When we talk about globular proteins, it tends to be more spherical or having weird looking shapes. It's a glob of amino acids pushed together. Fibrous proteins generally give me the structure of my cells or the structure of my body parts, where globular proteins do very specific functions. They're going to be immune reactions. They're going to be acting as an enzyme. Globular proteins do more specific jobs, whereas fibrous proteins build structures. Fibrous proteins don't play nice with water. They're not going to dissolve. They don't like to float around in water. Whereas globular proteins are a lot more comfortable being in water. And fibrous proteins are much more stable than globular proteins are. Remember that if I lose my 3D shape, I'm called denatured. Globular proteins are much more likely to denature than fibrous proteins are. So I've given you some examples of fibrous proteins, given you some examples of globular proteins, as well as some of the functions that they do. Notice that for the most part, the kinds of functions we typically think about for a protein are done by those globular proteins, but it's still important to have these functions as well with those fibrous proteins. So a brief summary for you of the different functions that proteins do in the body. Notice that this is color-coded. So my fibrous proteins have a box that's this darker purple color. Remember that fibrous proteins are long. These are the proteins that help with attachment, they help with contraction. My globular proteins were the ones that looked a little bit more like a blob. Those globular proteins do a lot of different things, like communication, antibodies, transporting things like oxygen, or acting as enzymes. So a good summary of some of that information from the previous slide. When we talk about proteins acting as an enzyme, remember that for chemical reactions to occur, many chemical reactions require what's called activation energy. Activation energy, as we see in our picture, is kind of like the hump that we have to get over to get things started. When I have enzymes, enzymes are going to act as a catalyst. And this word catalyst just means something that decreases the size of that energy hump. So the amount of activation energy required goes down because an enzyme's there. That's because enzymes can help create an environment that's better for a chemical reaction to happen. And if it takes less energy to start a reaction, that reaction can occur much more quickly. So enzymes end up increasing the speed that reactions happen at. So, highlighted here for you, and let's make sure we understand. Enzymes decrease the amount of energy required, and because of that, they increase how quickly these reactions occur. A big emphasis point, though, with enzymes, I use them over and over and over again. They won't be broken down, which is why we can constantly build new DNA. It's why we can constantly break down molecules in our body, because our enzymes are never used up. They're never destroyed. When we look at how enzymes work, they take two reactants, or another word that we talk about are substrates, and they're going to bring those substrates closer together and help to create a bond between them to make a product. As you can maybe tell from this picture here, enzymes have active sites that are very specific. So the only thing that would fit into these two spaces on my enzyme are amino acids. This enzyme can only ever build proteins because it's highly specific to work specifically on one substrate or on one reactant, in this case, amino acids. Many times when you look at the name of an enzyme, it'll tell you what it's building or what comes from it. So some examples of things like that include the enzyme peptidase, which helps to break down peptide bonds. Things like lipase, which breaks down lipids. Or we have an enzyme called DNA polymerase, and that helps to build DNA. So when you're looking at the name of an enzyme, you can probably tell its job just based on that name. On that note, DNA polymerase, let's chat a bit about the last kind of organic compound in the body, which is nucleic acids. When we talk about nucleic acids, they have a very similar chemistry to our proteins in that they do include nitrogen, but nucleic acids also have phosphate. So this is a new element that we haven't seen before, and that phosphate is going to be important to helping us form our DNA. So nucleic acids as a group, which is DNA and RNA, that's the polymer made of small monomers called nucleotides. These nucleotides are the little pieces. It's the letters that you've heard of before. So things like A and T, G and C. These are our nucleotides used to build DNA. A nucleotide has a nitrogenous base, which is the identity, if you will, of the nucleotide. It's attached to a pentose sugar. Pentose means there's 5 carbons, and there are 2 5-carbon sugars that we use to form nucleic acids. Their names are deoxyribose and ribose. So unsurprisingly, deoxyribose sugar is in DNA, ribose sugar is in RNA. So ribose sugar, ribonucleic acid, deoxyribose sugar, deoxyribonucleic acid. When we talk about nucleic acids, their big picture function is information storage. This is how we know what to make. This is how we know what we should be doing. Nucleic acids direct processes in the cell. The 2 types of nucleic acids are different from one another. We should be very familiar with this already from 1441, but a quick review between DNA and RNA. DNA is found in the nucleus of a cell. RNA starts in the nucleus, but it leaves the nucleus. So RNA not found in the nucleus all the time. DNA is also a double-stranded molecule, as we see here in our picture. RNA is a single-stranded molecule. Sometimes it folds on itself, but for the most part, we just see this single line, like we can see right here. The bases of DNA and RNA are not the same. When we talk about DNA, the 4 bases it uses are lined up here on the left. Adenine, base A, always pairs with thymine, base T. So A and T always go together. Guanine and cytosine always go together, G and C. But when we're in RNA, remember that we do not have the base thymine. Instead of thymine, we have a very similar base, though not identical, a very similar base called uracil. So A and U pair together in DNA. G and C, or in RNA, excuse me. G and C still pair together in RNA just like they did in DNA. We already mentioned this before, but we've got 2 different sugars in DNA compared to RNA. We also should consider that DNA is what I use to store information, whereas RNA is what I use to directly build things. This is why DNA stays in the nucleus of your cell, because it's storing the instructions. When I know which instructions I'm interested in using, I will send RNA out of the nucleus to make those processes happen. One other quick note about nucleic acids. Technically, we use a nucleic acid to build ATP. ATP, if we write its full name, is adenosine triphosphate. Adenosine as in I'm a modified adenine base. So ATP is being mentioned here with nucleic acids, just like it's been in other lectures, but specifically with nucleic acids because it has a nucleotide inside of it, because it has A, just like your DNA has. When I attach phosphates groups to it, now I can use it as my energy money in the cell, but it still starts as a base, and that base is the same as what we put in your DNA. On that note, I want to draw your attention to this activity here on the last slide of your notes. This is a table to help you organize the information from this lecture. I've either given you the name of the polymer that we talked about or the name of the monomer that we talked about. I want you to work on filling in the blanks on this table, either with the names of the polymers or the monomers. If you're able to sort that information out, you're in good shape and you understand your organic molecules. So good luck with that table and with your continued studying.